Finally, our program is being live captioned, and you can turn on the captioning in the menu, which is usually at the bottom right of the Zoom window. So with that, I'd like to introduce our speaker for today. We have Ian Gilman speaking for us. Ian is a PhD candidate in the Ecology and Evolutionary Biology Department at Yale. Originally from Pennsylvania, Ian has a Bachelor's of Science in Physics from Bucknell University and a Master's in Science in Biological Sciences from the University of Idaho. He joined Erica Edwards' lab at Yale in 2017. And Ian's work explores the evolutionary relationships of plants. His talk today will focus on how evolution by duplication has become a hallmark of flowering plants and how gene and genome duplication has impacted the evolution of photosynthesis. So with that, please welcome Ian Gilman.
All right, hopefully you can see those slides. Thank you so much for the
Ian,
introduction,
sorry,
Megan.
I can't see your slides. Can you try sharing your screen again?
Yeah. Better?
Looks great.
Great. All right. Well, thank you, Megan, so much for the introduction. And I'd like to thank the Peabody for giving me this awesome opportunity. And of course, I heard there's a lot of people joining us live today. So thank you all for being here to hear me talk about my research on photosynthesis and evolution by duplication. As you probably guessed from the flyer, I'm a botanist by training. I work with the flowering plants, also known as angiosperms. And the angiosperms are fascinating for a ton of reasons. Most apparent might be the beauty of the group. Everyone loves a nice flower, and it's a really easy way to get started doing plant research. But what you might not know, and what really interests me as an evolutionary biologist, is that the flowering plants have really remarkable genomes. So in animals like us, large genetic changes tend to cause disease or death, but in plants, they're a key feature of evolution. For example, if you go to the supermarket and pick up a strawberry, it'll actually contain about 8 copies of its genome, what's known as an octoploid. And the way that these extra copies arise is through a process called whole genome duplication. This typically happens through an error in meiosis preceding sexual reproduction. Normally, in sexual reproduction, parents each pass on 1 chromosome through their sperm and egg, which go on to form pairs in the progeny. But if, in error, each parent contributes both chromosomes, the progeny end up with both pairs from both parents. And this means that in just a single generation, we have an entire doubling of all of the genetic material. So although genome duplication events are really events, they happen in just a single generation, their evolutionary consequences play out over many millions of years. Here's a diagram of the relationship between many major groups of plants extant today, called a phylogeny. And you can see species around today here on the right, and then we can trace their relationships like a family tree back in time to the left. We can see, as indicated by these colored bubbles along this phylogeny, that there have been a bunch of whole genome duplication events in the past, leaving many of today's species with 2 or more copies of the genome, like in the strawberry. And these duplication events have led to many of the most amazing evolutionary innovations in the plant tree of life, such as the flower itself, which evolved through the duplication of Madsbox genes that give rise to the different parts of the flower, like the petal, sepal, stamens, and carpels. And even more recently in time, the evolution of glucosinolates in the mustards have arisen through genome duplication. And glucosinolates are those spicy compounds we enjoy in things like horseradish, wasabi, and arugula. But the focus of today's talk will be on the role of genome duplication events in the evolution of photosynthesis and how these duplications have allowed plants to thrive in really extreme environments. But before I go any further, I'd like to give a little primer on genes and genomes so we're all on the same page for the rest of the talk. When I say genome, I'm referring specifically to an organism's nuclear genome. That's all of the DNA housed in our cell nuclei. In humans, this is about 3 billion base pairs organized into our 23 pairs of chromosomes. Our cells have other genomes, though, like in our mitochondria, and plants have a third genome in their chloroplasts. But again, today we'll just be talking about nuclear genomes. And the genome is best understood as a sort of blueprint, which contains all of the plans to build the proteins and enzymes that actually go out and perform roles in our cells. And this blueprint is read at the level of individual genes. Here's an example of a single gene and how that information is transformed into a protein. As this diagram probably conveys, genes are pretty complex. And that's because they not only code for the information inside the protein, but also all of the instructions for how and when to make it. So if we want to make a particular protein, some molecular machinery first has to find this gene and figure out which direction to read it in. So in front of every gene, there's a little promoter sequence right here highlighted in pink, which does exactly this. And then inside the gene itself, we have two types of sequences: exons, which end up becoming protein, and introns, which contain other little bits of instructions and need to be removed before we make our protein from those intermediate mRNAs. Putting it all together, genes need to be so complicated because they again code for the protein itself, as well as all the instructions that tell the molecular machinery when to use the gene and how to build that final protein. And most species have between 30 and 60,000 genes in their genomes, many even more complicated than the one we're looking at here. And I think that this raises a really interesting question, because if even a single gene is so complicated, how is it that we ended up with tens of thousands of them in all of our genomes? Especially if we think of evolution as this very slow, steady, and random process of just single letter changes in the genetic code. Well, as you may have guessed from the introduction to the talk, a lot of this evolution proceeds via duplication. As Dr. Susumu Ohno put it, nothing in evolution is created de novo. Dr. Ohno is responsible for bringing evolution by gene duplication to the forefront of evolutionary biologists' minds through his 1970 book, Evolution by Gene Duplication, where he outlined how gene and genome duplication occur and some of the remarkable innovations that have resulted from them. So let's think about what can happen following the duplication of a gene. Here, I'm showing you a simple protein with two domains. That is, it has two little parts to it here in orange and pink. And we can imagine these two domains being coded for by two exons, again, in this gene highlighted by the same colors. Following a gene duplication event, a few things can happen. The first might be simple conservation, where both copies of the gene retain that ancestral role. Maybe they make more product of this gene, maybe they don't. But in general, this is pretty simple. A few more complicated scenarios are possible, though, and these get really interesting. The first is subfunctionalization. And this is where each copy of this duplicated gene evolved to perform only a subset of the original gene functions. What this can allow is separate evolution of each of these domains in ways they might not be able to when joined together. And this might also allow for differential use of these two copies, perhaps changing the ratios at which the domains are produced. We can also have specialization occur, which is when both copies, for the most part, retain that original function, but each evolves for a slightly specialized use. For example, this might have been a protein that originally was sort of a jack of all trades protein that was used throughout our body, but separate evolution might allow each copy to be more optimally tuned for the temperature or pH of different tissues, or perhaps to be used in different amounts at different times or in different places. Finally, and perhaps most interestingly, is neo-functionalization. This is where one copy retains that ancestral role, but the other one begins to take on a new function entirely. And it's neo-functionalization that we're going to talk more about, because it's played a big role in the duplications that facilitated plant adaptation to a low CO2 world. And as humans, we might not think that we live in a CO2 world. We actually are living in a very high CO2 world relative to our time on this planet. And we're scrambling to reduce the effects of climate change as CO2 levels rise exponentially. But plants have been around for hundreds of millions of years. And for most of that time, CO2 was 10 to 50 times what it is today, which was great for photosynthesis. Today, photosynthesis is up to 30% less efficient in a lot of plants simply because CO2 is so low in the atmosphere. And this is because plants actually obtain all of their CO2 from the air passively in exchange for water. When plants take up water from their roots, it immediately moves the entire length of the plant and exits through the leaf out of these little pores called stomata in the form of water vapor. In exchange for water vapor, CO2 is taken up through these same little pores. And at the current exchange rate, plants have to give up about 400 molecules of water to gain 1 molecule of CO2. And this means that plants spend over 95% of their water just taking up CO2. Of course, evolution has risen to the challenges of a low CO2 world in the form of adaptations we call carbon concentrating mechanisms. The most common of which are called C4 and CAM photosynthesis, and these allow plants to live in extreme environments, like up in the rainforest canopy, in nutrient-poor aquatic habitats, deserts, and we also find them in some of our most productive crop species, like sugarcane and maize. Plants without carbon concentrating mechanisms do what we call C3 photosynthesis, and this is a relatively simple process where CO2 again enters the leaf through those little pores and immediately enters the chloroplast, where the enzyme rubisco helps transform it into sugar. In our first carbon concentrating mechanism, called C4, we see that we have a 2-cell type of photosynthesis. Here, again, CO2 comes into the leaf during the day, but it's now transformed into a 4-carbon acid, shown here in black. This occurs in the mesophyll cell, and then these acids are moved into a second cell called the bundle sheath, where they're broken apart again to release CO2. Once this CO2 is released, rubisco can again begin making sugars. And by using 2 cells for photosynthesis, these plants are effectively tricking rubisco into thinking it still lives in a high CO2 world. And this happens because the conversion of CO2 in the mesophyll generates a really strong diffusion gradient for CO2 to enter the leaf. And then the rapid release of CO2 in the bundle sheath concentrates CO2 for rubisco at levels not seen for about 10 or 50 million years. CAM, on the other hand, is still a 1-cell system. But what's unique about CAM is that these plants do all of their gas exchange at night. And by taking up CO2 at night, when temperatures are lower and humidity is higher, these plants save a lot of water. As in C4, CO2 is converted into a 4-carbon acid here in black, but then these acids are stored in an organ called the vacuole overnight. During the day, the stomata close, stopping gas exchange and preserving water during the hottest, driest parts of the day. Then the vacuole releases stored acids, and these are again broken up, re-releasing CO2 at extremely high levels. And again, because plants are doing their gas exchange at night, they're saving 7 to 10 times the amount of water that non-CAM plants spend to gain carbon. So if you're paying really close attention to these diagrams, you may have noticed that here in orange, that C4 and CAM are sharing a set of metabolic reactions. And they don't just share these metabolic reactions with each other, they share those with all plants, and in fact, us too. Because these reactions are anaplerotic reactions. They're metabolic intermediates needed for essential processes in all organisms that respire. These are all the organisms like us that basically break up the energy or break up food that's stored in our bodies to release energy. And so like us, plants at night when the sun isn't shining need to get their energy by breaking up stored carbohydrates. And so what's happened in the evolution of C4 and CAM is that the genes responsible for these anaplerotic reactions have been duplicated and then neo-functionalized for a new role in C4 and CAM. And what's really cool about the evolution of C4 and CAM is that they rely on the duplication of very common genes, these anaplerotic reactions. And because plants in general have a lot of genome duplication events, most plants around today have the building blocks to evolve carbon concentrating mechanisms lying nascent in their genomes. And in fact, if we look across the plant tree of life, we see that C4 and CAM have evolved dozens of times independently in a phenomenon called convergent evolution, where different species have converged on the same evolutionary solution to the common problem of low CO2. And here's a few examples of some of the plants that use C4 and CAM, like air plants you might have in your home, orchids, pineapples, and a lot of our desert giants. And then C4 plants include a lot of crops like amaranth, sugarcane, and maize. But for the rest of the talk, I want to focus on my favorite group of plants, which are Portulaca, the purslines. These little amazing plants are rare in that they don't just have one carbon concentrating mechanism, but they're able to do both C4 and CAM together. They perform C4 every day, but then they additionally use CAM when stressed. And I know all of you are at least a little familiar with Portulaca because this species, Portulaca oleracea, is one of the most common weeds found in every country on the planet. So before you pull this weed out of your sidewalks and driveways next summer, just remember that it has one of the rarest and most efficient forms of photosynthesis that we're aware of. My research in Portulaca is mainly focused on the coordination of C4 and CAM, again, because it is such a rare phenomenon. We're interested in knowing if the same genes are used for both metabolisms, or if there were perhaps additional duplications that allowed for different genes to be used independently in each pathway. We're also interested in understanding whether or not all Portulaca, there's about 150 species of them, have combined C4 and CAM in the same way, or if perhaps there's different ways to sort of plug these metabolisms together. The way that we addressed these questions was by measuring gene activity while plants transitioned from a C4 only state to a CAM state. And we studied this in 2 species. This is a relatively easy experiment to do. We simply grow these plants up under normal conditions where they're nice and happy and well watered, and then we just stop watering them for about a week or 10 days. Over the period of this experiment, we monitor the acid levels in leaves, and we're looking for a fluctuation in acid levels between the day and the night. And remember, CAM plants concentrate acids in their leaves overnight to be used during the day. And so when we see this cyclical acid induction, we know that the plants are doing CAM. Throughout this experiment, we're measuring gene activity for tens of thousands of genes 4 times every day, as indicated by these little DNA markers shown on these plots. We then correlate all of this gene activity over the course of our experiments for those tens of thousands of genes, and we use those correlations to build networks of potentially interacting genes. And we constructed 4 of these networks, 2 for each of our species, once when they're doing C4 only, and then when they're doing C4 and CAM together. The details of these networks aren't important for the talk today, but I want you to know that by comparing these networks between and within species, we're able to identify which genes are involved in C4 and CAM specifically. So we were able to take that information in our gene networks and translate them into these schematics of how photosynthesis works day to day in these 2 species. Again, the details aren't important, but we were pretty successful in describing these metabolisms in detail when plants are doing C4 only, and then when they transition to using C4 and CAM together. Here's a simplified diagram of how C4 and CAM are coordinated in Portulaca. In Portulaca, you can see on the right during the day that C4 proceeds normally, with acids made in the mesophyll transported to the bundle sheath cell, where they're broken up and used by rubisco. And at night, CAM also proceeds pretty much as expected. But during the day, what happens is that the acids generated in CAM are released and allowed to join with the acids created in C4. And all of them are jointly transported to the bundle sheath cell to be broken up, release CO2 to be used by Rubisco. What we've learned with our gene networks is that it is the case that different gene copies have been used in C4 and CAM. So for every gene that we see here used in CAM in pink, we found at least one copy that's distinct used in C4. And we only found evidence for perhaps one gene that's shared by C4 and CAM. And by comparing our two species, we found out that there were slight different biochemical differences that suggest that there are more than one way that C4 and CAM are combined. So they both fit that general framework of how C4 and CAM are coordinated, but there are slight differences in their biochemistry that suggest that there are nuances to how these metabolisms can be combined. Along the way, we made a couple other interesting findings, such as a single point mutation, that is just a single letter change in one protein that we think facilitates the coordination of C4 and CAM. And we also found evidence that CAM evolved by linking some of these duplicated genes to the circadian clock. We studied this last point by looking at all of the genes used in CAM in our networks. We then looked at all of these genes in this network in the Portulaca amylase genome, which we sequenced as part of this project. Specifically, we looked at the promoter sequences of all of these genes. And if you can remember back to those first few slides, the promoter sequence is critical in specifying when a gene needs to be used. So by comparing promoter sequences, we're searching for common signals that might coordinate the use of the genes in this network. And just like animals, plants have circadian clocks that dictate when things occur in their cells and bodies throughout the day. And our comparisons revealed that the promoter sequences of these genes in the CAM network are read by the circadian clock. And this made a lot of sense to us because CAM is a highly time-structured metabolism. It relies on the opening of those pores at night for gas exchange and then nocturnal conversion of CO2 into these acids. Then those acids need to be stored overnight. And before those acids are allowed to be released, gas exchange has to halt. Once gas exchange stops, those acids can be released and broken apart and release CO2 for the plant to use again. So it made a lot of sense to us that the evolution of CAM, this highly time-structured thing, would occur by linking into existing time-structured signals in the plant. Through our work and that of others, a detailed understanding of photosynthetic evolution in Portulaca is emerging. Here I'm showing you another phylogeny. This one is of Portulaca there at the bottom, and a lot of its close relatives, like the cacti shown by that saguaro, and the spring beauties up there with that little herbaceous plant. We can see relatively deep in the history of this group, a genome duplication event shown by that star. It's shared by most of Portulaca's relatives. Following that genome duplication event is where we think CAM evolved, by linking some of these duplicated genes to the circadian clock. We think that CAM evolved here because all of the relatives that share this genome duplication event are also capable of CAM and use the same gene copies as Portulaca do. Then on the way to Portulaca here on this phylogeny, in orange, we can see over 500 more gene duplication events. And we think that these gene duplication events are what facilitated C4 evolution. And we believe that C4 has evolved actually 3 times independently in Portulaca because of those biochemical differences that we found when comparing Portulaca oleracea and amylase. There's still a lot more to investigate in this group, and so I'm going to be continuing to study Portulaca this summer in a postdoctoral research position at Michigan State University with the National Science Foundation. And some of the questions that we're looking to tackle in the projects we've designed are how how genes become restricted to particular cells. We know that C4 relies on the evolution of cell-specific gene expression. That is, we have to have some cells working in the mesophyll only and others only in the bundle sheath cells. But we're not sure how this evolves. We also aren't sure what the molecular signals are that connect stress and CAM. We know that CAM has evolved with the circadian clock, but we aren't sure what the cascades of molecular signals are that allow plants to stress something like a drought signal and then begin using CAM in addition to C4. So with that, I would just like to thank the funding sources for my research and the Peabody for this great opportunity. And of course, none of this research would be possible by myself. I work in a lab that has a lot of really great graduate students and postdocs, led by Dr. Erica Edwards. And other labs that have been crucial in this work include that of Carolina Hayduk at UConn, Eric Goolsby at Central Florida, and Dr. Payat Christian. And nothing in our lab gets done without Christopher Bullock at Yale's Marsh Botanic Gardens and at the Yale Science Building, who runs all of the greenhouse and growth facilities for research. So with that, I will take any questions.
Great. Thank you so much, Ian. That was a really interesting talk. As a plant ecologist myself, I found it fascinating to focus on the genome, which is something I don't focus on much in my research. And I wanted to congratulate you on your postdoc position.
Thank you.
Yeah. So I just wanted to start off with a really general question, more for you. And that's what was your entry point into plant genetics and how did you become interested in this research topic?
Yeah, that's a really interesting question because I did start off my science career as a physicist. And I think that a few things kind of met all at the same time. None of it really planned, but in my, I don't know, sophomore year, junior year of my undergraduate, I was taking a biophysics course. And a new botanist started working at Bucknell University named Chris Martin. And so at the same time, I started taking an introductory botany course and a biophysics course. And I realized that I could start applying some of the things I was doing in physics, like a lot of computational methods and a lot of statistical things, but not look at things like black hole gravitation or the movement of planets. And I could apply them to things like plant research and plant genetics. And I had a lot of fun working with mating systems in tomatoes in a greenhouse as an undergraduate. And that led me into field botany. And after that, it was just one thing after another with plant research.
Great, thanks. I love hearing about the evolution of how you came to study what you do in this field. I'm going to start with a question from an audience member. John asks, does the combination of C4 and CAM only occur in flowering species, or can this also occur in other organisms, for example, in algae?
That is a good question. And we only know of C4 and CAM from vascular plants. So those include the flowering plants, but also things like conifers and ferns, things like mosses, which don't have true vascular systems. We don't see C4 and CAM in. And then we do see CAM actually in the lycophytes. So if you know the plant Isoetes, it's kind of a rare and strange plant that lives in submerged aquatic habitats. So we do know of them from non-flowering plants, but once you get into things like algae, they do things like bicarbonate concentration. So they'll use a different carbon concentrating mechanism that directly uses bicarbonate. But we don't know of any that use C4 or CAM. And I think a thing that's harder to imagine with algae is C4, which is this two-cell system where a lot of algae are single cell or have really rudimentary multicellularity. Yeah.
Okay, thank you so much. I want to go to a question from William that says, could you go back and give a little bit more insight into the historic change in carbon dioxide exchange, and photosynthesis. I think this is about the CO2 in the atmosphere, and how that shifted over time.
Yeah, this is really exciting. I have another project as part of my dissertation looking at how changes in atmospheric CO2 have led to the evolution of C4 and CAM. So we think that C4 and CAM have both evolved in response to this drop in CO2, beginning about 30 to 50 million years ago, right when kind of there is this crossover from very high in the thousands of parts per million CO2 to below about 500 parts per million CO2. And I didn't go over it today, but it's not just a water problem for plants, but when there's less CO2 in the atmosphere, the enzyme ribisco actually has this long name that makes it a carboxylase, can act on carbon, but it can also, or CO2, but it can also act as an oxygenase with O2. And when it acts on O2, we get a series of energetically costly reactions that make plants actually spend a carbon atom and additional energy just to kind of get rid of some messy or messy intermediates that are a little toxic to the plant. And so as CO2 has decreased over time, plants have both less food in the air in the form of CO2, but they also have more oxygen and less CO2. So they start doing this additional oxygenase behavior at a higher rate too. So they're kind of getting hit on two sides. And those things together have led to the evolution of carbon concentrating mechanisms. We think because there is this critical point, about 500 parts per million, under which photosynthesis started becoming a little bit too inefficient. So who knows, as we continue to pump CO2 into the atmosphere, plants will probably be a lot happier, even if maybe we're not.
Okay, gotcha. Thank you so much for going more into that. We have another question from John that asks, can gene duplication be accomplished artificially, for example, using CRISPR?
Yeah, definitely. CRISPR generally works by us going in and we can make insertions of a whole new gene. So if we can put in a whole new gene, we could definitely duplicate an existing one. I have a project primarily led by Vivian Irish's lab that works with a postdoc in her lab, Adam Saffer, also on trying to get a CRISPR system working in portulaca. We've had minor success, but haven't really got it working yet. But we're looking to figure out if we can, again, kind of tinker with the genetics to further understand how these systems are coordinated. And I think that very far down the line, our work might be applicable to things like trying to put in some of these duplicate genes to maybe get water use efficiency increased in existing crops or things like that.
Okay, cool. So then that could have some impacts for climate change and agriculture that's impacted by drought, for instance.
Exactly. Yeah. The main benefit would, we think, trying to get something like CAM into already C4 species, trying to get them to be a little bit more water use efficient than they already are.
Yeah, that's really neat. That's really cool to make that link between the sort of, I feel like we talked a lot about the genetic background of this work, and it's nice to make that connection to how it could be applied sort of to deal with problems that are existing that might be affecting us more and more in the future. So thank you. We have another question from Sydney who asks, is portulaca's dual photosynthetic mechanism part of what allows it to become such a successful weed?
Yeah, we think so, but it's hard to show that. So the mechanism is pretty rare, but there's about 150 species of portulaca, and all of them can do this. And so only one of them is really such a big weed, which kind of begs the question about why the other ones aren't. But it certainly seems like it should be a major part of why they're able to do this. And if you find portulaca, they're often growing where no other plants are. So to me, they seem like not very good competers with other species. They don't really pop up in your lawn. They pop up most in these sidewalk cracks and driveways in really dry places that really don't seem like they should have any soil. They can't hold on to any moisture. And they can also complete their life cycle really, really quickly. So the plant can go from a seed to producing seed in just about 6 weeks. So it seems like there are a few traits that are all packed into portulaca that make it a really good weed. Um, and it seems like this has to be one of them.
Okay, cool. That's really interesting. Thank you. I was wondering if you could tell us a little bit more about the stress experiments when you were measuring the gene activity in the plants. It sounds like you did those in the Marsh Botanical Gardens, and I was just wondering like what that looks like, and how you monitor those plants, how long those experiments are going on for you to get this type of information.
Yeah, these are pretty labor intensive. And so I think I've run maybe 4 or 5 of these over the course of my time at Yale. And they have all been done actually not down at the Marsh Botanic Garden, but they're all done in very highly controlled growth chambers that we can control the temperature and humidity and even CO2 level of these little growth chambers that are about the size of maybe 2 or 3 fridges large. And we can control things like the light level. And so we could do this with very, very high precision. We grow these plants in these conditions. And then we measure photosynthesis in a few ways. So we have a machine called a Lycor, which you may have heard about in some of the other ecological talks we've had, where we can basically take this box and stick a plant inside of it, and we can measure in real time them doing photosynthesis, actually exchanging CO2 with the atmosphere, and whether or not their stomata are open, is water coming out of their stomata, things like that. So we can really measure these plants, what they're doing photosynthetically second by second. And so we have things like that to monitor their physiology. And then we grow these plants up for weeks in these conditions. And then we go in at these 4 times of the day. And you probably don't realize it when it passes you by quickly, but these time points are at 4 a.m., 10 a.m., 4 p.m., and then at 8 p.m. again. So it involves like sleeping in the lab. It's not a lot of time off because staggered with that too, we're measuring different parts of the physiology. And so it really is kind of like an all hands on deck. You're in the lab for about a 48-hour period. So,
wow.
Yeah.
Yeah, that sounds really intensive. And you said you did how many of these experiments during your PhD? 4 or 5?
I think I've done 4 or 5 of these. Not all of them were successful. Sometimes we had problems with little malfunctions in the growth chamber where lights would click on in the middle of the night.
Oh.
And then actually the one that I'm showing you data from today, actually the experiment ended about 4 hours before Klein Tower had that fire a few years ago. And
Oh
so
my
we
goodness.
were like just out of the nick in time, got this experiment finished up. So yeah, these data are pretty important to me.
Yeah, they sound very precious. I'm glad we got to see them today. Yeah, I had one last question for you, which was, well, so you were talking about measuring, these photosynthetic processes in the lab, and I was wondering if you measured any other processes, or it sounds like you can maybe measure some different processes with the equipment that you have to do the lab work.
Yeah, I would say that they do sort of separate things. So when we're looking for these genes, we don't know what their roles are. We need some sort of ground truth to say, what are these genes doing? Are they actually involved in the processes we think that we're observing? And so by measuring these parts of photosynthesis, like the changes in leaf acidity and whether or not their stomata are opening or closing, are they doing gas exchange? We're basically providing the physiological data that confirms the metabolisms that we think we're looking at. And then that allows us to kind of hold up the genes next to it and say, yes, these genes and the changes in gene activity over time are correlated with the metabolisms that we're or physiologies that we're measuring in the with other metrics. So they are complementary.
Okay, cool. And then, so it sounds like, I guess what you're saying is that some other plant processes can be affected by gene duplication. And then these types of experiments sort of help you sort out which ones are being particularly affected by specific gene duplication.
Exactly. Yeah. So if you look at these networks, for example, they're huge messes with tens of thousands of genes in that. And so the way that we're able to pare down that giant mess of interactions to just those involved with the things we're interested in is by basing them on those physiologies that we're actually observing in the lab, because otherwise you just have this mess of 45,000 genes all kind of going in different directions. And so that physiology in the lab lets us ground ourselves.
Okay, great. Well, thank you so much for describing all of that for us. Thanks so much for showing us these beautiful messes of gene networks. And I do think we're about at the end of our time today for this talk. I wanted to thank you for being here, Ian, and thank all of our audience members for being here. You can look out for our next talk in this Research Spotlight series from Kelsey Jenkins, which will be taking place on March 30th. And Kelsey is a PhD candidate in the Department of Earth and Planetary Sciences, and she'll be telling us about vertebrate fossils. In the meantime, if you're out in the audience, please follow us on social media. You can stay in touch by signing up for the mailing list on the Peabody website as well. And we'd just like to make sure you're able to catch all of our online and in-person programs this semester and beyond. I hope everyone has a wonderful rest of the day. Again, thank you to Ian, and thank you everyone for being here. Have a great Thursday.